Good morning. Thank you for introducing me. I want to thank the organizer for inviting me and give me the opportunity to present my work here in these conferences. Of course, the title of my presentation is Phytates from Bifidobacteria for Human Nutrition. This work was developed in the cereal group of the Department of Food Science of the Institute of Agrochemistry and Food Technology here in Valencia. As probably everybody knows, the myo-inositol hexaphosphate or phytic acid and phytates are the primary source of inositol and the major storage form of phosphorus in plant. They are present in seeds, cereal grains, and legumes. So the chemical structure of phytic acid is shown in the pictures. It is the myo-inositol ring is a polyalcohol, cyclic polyalcohol, phosphorylated with 6-phosphate. This molecule is negatively charged, so can bind a lot, all of minerals like calcium, zinc, iron, magnesium, and prevent their uptake. Also forms complex with proteins, amino acids, and also inhibit the activity of some digestive enzymes. So in the human and animal nutrition, the phytic acid could be included as anti-nutrient factor. The strategies for reducing or eliminating phytates are in food or feed are, for example, the change in food production processes, the selection of varieties with high phytase activity, genetic engineering, changes in agricultural conditions, addition of exogenous of phytases, or inclusion of phytate-degrading microorganisms. The most effective strategy is to include an exogenous phytases during process. In general, they are from bacteria or from fungi. The phytase is a phosphomonoesterase capable to hydrolyze the phytic acid to free inorganic phosphate and lower myo-inositol phosphate esters. As the number of phosphate groups are progressively removed from the phytate, the mineral binding strength decreases and solubility increases, forming weaker mineral complexes. It's important to remark that the structure of phytic acid is a chair structure with one phosphate in axial position and the rest of phosphate in equatorial position. This is important to understand because depending on the phytase, the first phosphate attack could be in the position 3, 4, or 5. This first hydrolyzed phosphate group can vary in position, depend on the enzyme, and subsequently the hydrolysis will continue on another specific group of the inositol ring, so generating a particular hydrolytic profile for each enzyme. So what was our idea? To include a strain, a starter phytates producer. So we collaborate with the Institute of Animal Reproduction and Food Research of the Polish Academy of Science in Olsztyn, Poland, in the microbiology laboratory. We looked for a strain from the genus Lactobacillus, phytase producer. We checked a lot of strains, but in the bibliography and also in all the strains that we checked, more than 100, we didn't find phytase activity in Lactobacillus. Lactobacillus, the strain of this genus don't produce phytase. So we thought that it could be interesting to include bifidobacteria in the bread making process. So first we checked the phytase activity in this genus. We checked many strains also of bifidobacteria, and Why bifidobacteria? Because they are main source of probiotics, so we can include them in the process because they are qualified presumption of safety, like lactobacillus. So we only found phytase activity in the species longum, subspecies longum, longum subspecies infantis, pseudocatenolatum, and dentium. In the next picture, you can see different chromatograms with the main peak is the phytic acid. The first chromatogram is the control, is a culture medium without inoculation any strain. So we can see the peak of phytic acid. In the second chromatograph is the culture medium with a strain, no producer phytase, like Bifidobacterium breve. And the third one is Bifidobacterium longum subspecies infantis. After 12 hours of incubation, the phytic acid disappeared completely, and the IP3, the myo-inositol 3-phosphate, was accumulated. We also study how the expression of these phytases change with the, in different mediums. For example, if free phosphate is present in the medium, the production of phytase is lower. But if the bifidobacteria grows in a medium with a prebiotic like phosphooligosaccharides, the expression of this phytase is higher and hydrolyze the phytic acid quicker, faster. So we wanted to understand which is the pathway of the hydrolyze of this enzyme from bifidobacteria, because the pathway is important to understand which kind of lower myo-inositol phosphate generate this enzyme. Why? Because some of them have important biological functions in our body, like anti-inflammatory activities or maybe antioxidant activities, increased mineral availability, and so on. So we collaborate with the Food Science Division of the Department of Chemistry and Bioscience of the Chalmers University in Gothenburg, Sweden, for this purpose. So in this picture, we can see two chromatograms. We use HPAC. So the first one is the phytic acid hydrolyzed by chlorhydric acid. This is a chemical hydrolysis. So in this chromatogram, we can see all the lower myo-inositol phosphate, because depending on the position of the phosphate group which was hydrolyzed, is the profile of the hydrolysis is different. So because the one group phosphate is in axial position in the second carbon and the rest in equatorial position. So depending on the hydrolysis, we have one isomer to another one. So in the second chromatogram, we can see the hydrolysis by Bifidobacterium pseudocatenolatum phytase producers. So we saw that this bacteria can hydrolyze the 6th position and the 5th position. That is new in the world of phytases, because phytases can hydrolyze the 5th position or 6th position or the 3rd position, but not those two positions. So at the beginning we thought that Bifidobacteria produce two different enzymes. Because the 6 and the 5 positions as the first step of the hydrolysis, the main isomers producer is the 1, 2, 3, 4 tetrakis phosphate, myo-inositol tetrakis phosphate, with the group 1, 2, 3. So the pathway of the enzyme, this enzyme, are two. Here in these isomers is missing the position 5, and here is missing the position 6. So generate these two IP4 and different IP3, which have biological functions. And another thing to remark here is that usually the enzymes hydrolyze up to the IP1. But this enzyme stops the reaction in IP3. So this is interesting in nutrition, for example, or in functional characteristics. Is another thing to remark, because these enzymes are different. There are many phytates in the market, but they are for animal feed. But there are not enzymes which produce the 1,2,3-triphosphate grouping. This triphosphate grouping is 1,2,3 is equatorial, axial, equatorial. So has the configuration that uniquely provides a specific interaction with iron. And this has been reported to be present in normal mammalian cells at high concentration and also increase its solubility. Of course, this grouping 1, 2, 3 is also in the phytic acid and the phytate, but the complex with iron are insoluble in our intestinal tract. So if the myo-inositol ring has only 3 phosphate group, the interaction with iron is a very stable complex, but it's soluble. So now our cells can absorb it in our body, can absorb them. This in the bibliography is described that it has a role in iron chelation and as an intracellular iron transport agent and or cellular antioxidant, and is also potential candidate for enhanced mineral absorption. So now we have the bifidobacteria phytase producers. So now we can include them in the process, in the bread making process. Of course, the enzymatic phytate degradation in dogs depends on many factors, like a fermentation time, temperature, pH, water activity, flour extraction rate, starter culture, mineral content, additive, and the bread making process. So our strength stop the hydrolysis in IP3, start from phytate and stop in IP3. We made bread with 2 different processes, the direct process and the indirect process. In our formulation, we only include flour, water, yeast, and salt only. And we include bifidobacteria phytase producer during the mixing. In the indirect process, we made a pre-fermented dough 24 hours before and include in the formulation with the rest of the flours, water, yeast, and salt. We fermented the dough and with controller parameters of temperature and humidity and baking. You know that bifidobacteria produce short-chain fatty acid. It's mainly lactic acid and acetic acid, and in a low concentration of succinic and formic acid, so they could be involved in lowering of glycemic index, and also the decreasing of pH can activate the endogenous phytase of cereals, so we can improve the mineral availability. And of course, it prolongs the shelf life. In the next slide, we can see the different breads. This one is the control with different amount of sourdough, 5, 10, 15, and 20%. So we can see that bread inoculated with bifidobacteria had similar technological quality as the control sample in terms of crumb and crust color, crumb texture, crumb structure, specific bread volumes, slice shape, and sensory analysis. This is important because the inclusion of bifidobacteria doesn't change the technological quality and the sensory quality. We also make a sourdough with rye bread in different proportions. In this direction, we can see zero addition of sourdough, 10 and 20%. And in this direction, different levels of whole rye flours. So we can see that the technological quality was affected only with the whole rye, the proportion of whole rye flours, but not with the addition of sourdough. That is important from the technological point of view. Also another formulation with whole rye bread and whole wheat flours, the same behavior. The sourdough didn't affect the technological quality and also the sensory characteristics. But what happened with the phytic acid? When we increase the concentration of sourdough inoculated with bifidobacteria, the phytic acid decreased significantly and increased the lower myo-inositol phosphate increase. The iron dialysability was increased at all. We check in the control samples, in the control sample, positive control with the inclusion of phytates, commercial phytates, and with 10 and 20% of sourdough, so the dialysability of iron increased significantly. There is a parameter, is a molar ratio between the phytate and minerals. In this case, we did it with iron, and when this parameter, the molar ratio, is higher than 1, predicts the lower absorption of iron in human nutrition. So our formulation had showed higher ratios than 1, even with 20% of sourdough. This prediction is that it's not enough, the addition of bifidobacteria phytase producer. And in fact, the ferritin, the effect of the treatment on molecular ferritin formation in CACO2 cells exposes to the gastrointestinal digestion of bread show that there are not significant differences between formulations. Now I will show you the direct process, bread made with direct process. We made the control, of course, a positive control with fungal phytase and with bifidobacteria. Bread inoculated with bifidobacteria had similar technological quality as the control sampler or bread with fungal phytase. In terms of crumb and crust color, crumb texture, crumb structure, specific bread volume, slice shape, and sensory analysis. What happened with the phytate? The inclusion of bifidobacteria in the direct process without sourdough directly in the mixer decreased significantly the level of IP6 phytate in the formulation 100% of whole wheat flour. In the formulation of 50% of whole wheat flours also have this decreasement, but in the same way than fungal phytase. When the level of phytates is higher, the bifidobacteria couldn't decrease efficiently the level of phytate. But we saw, if we compare the lower myo-inositol phosphate, we can see that the inclusion of bifidobacteria, even in the 50% of whole wheat flour, remain in a high concentration, the IP3, but is not the case in the sample included with fungal phytase. The iron dialysability increased also with the fungal phytase, but more with bifidobacterium phytates. And the molar ratio, which predicts the iron availability, is still higher than 1. So this prediction predicts that the iron will be not absorbed from our gut. In fact, the sample included with fungal phytates is lower than 1. So in the concentration of ferritin in Cactus cells exposed to gastrointestinal digestion of bread show that the ferritin in Cactus cells exposed with the sample with fungal phytates, which are lower than 1 in the molar ratio, is significantly higher than the control. But the bifidobacteria, they show the same behavior, like control. So we understand that to include the bifidobacteria phytase producer is not enough for removing the phytate at a level that is important for mineral availability. So we decided to isolate them, but the problem was that we spent many months to isolate the enzyme, but we couldn't take in the poor solution, because the enzyme, we understood that is binding to the membrane of the bifidobacteria. So we express the gene of the phytase of bifidobacteria in another host, and we could isolate the enzyme in soluble form and in pure conditions. And we include this in the elaboration on infant cereals, in the bread making process, and in fermented soy milk. In the elaboration of infant cereals, there is a step which is with alpha amylase treatment, which is the dextrinization of starch. In this step, the conditions are 55 degrees, 20 minutes at pH 5.5. That is the best condition for phytase from bifidobacteria. They are the optimal condition. So we include with the alpha amylase the phytase from bifidobacteria. And we compare the level of the reminded phytic acid in the product, compared with the control sample, of course, and with the commercial phytase. And at the cell level, we include the enzyme in the same level of activity. And we saw that the bifidobacterium, the phytases from bifidobacteria are more effective, like the commercial phytases, and because they are very specific to phytate. The commercial phytase, they can hydrolyze different substrates, but the phytases from bifidobacteria only hydrolyze the phytate. So they are very efficient, and they are really very different comparing to the other phytases. We test two formulation, multi-cereals and gluten-free cereals, and we saw the same behavior. The bifidobacteria can hydrolyze efficiently the phytate of these formulations. When we include the phytases in different formulations with white bread, whole wheat bread, and bread with 25% of whole amaranth flour and 50% of whole amaranth flour. The amaranth is a pseudo-cereal. Okay. The amaranth is a pseudo-cereal that has really high concentrations of minerals. You know that the recommendation is to intake whole grains, whole flour, so because they have a lot of there are a lot of vitamins, there are bioactive compounds, and high concentration of minerals. Amaranth has 3 or 5 times more concentration of minerals like whole wheat flour. But when the minerals are really high, the phytates are also really very high. So it's important to hydrolyze it. with the long fermentation or for including phytates. So the inclusion of phytates didn't affect the technological quality. They look the same, even increase the volume of the bread, and also hydrolyze effectively the phytates, more than the commercial phytates in all the formulations. And in soy milk, we include the lactobacillic acid expressing bifidobacterial phytates, and also we have the same behavior. The control sample didn't modify the phytic acid during fermentation, but with the strain with the phytates of pseudocatenolatum decreased immediately the phytic acid and accumulate AP3. And Longus subspecies infantis also have the same behavior, but then not so fast.
Excuse me, doctor, we need to go ahead and wrap up, please, for the next speakers.
Okay. As conclusion, the phytates from Bifidobacterium longum and pseudocatenolatum can be categorized into the class of histidine acid phosphatases, which are active at acidic pH and have histidine on the active site. They present the first phytases characterized from this group of probiotic microorganisms, opening the possibilities for their use in the processing of high phytase content products for human or animal nutrition. The phytate degrading activity constitutes a novel metabolic trait which could contribute to the improvement of mineral absorption in the intestine as a nutritional probiotic future with potential trophic effect in human gut. Thank you for your attention.